Hello everyone, welcome to Tawhid Foundation for Allied Healthcare Sciences, Bangalore. Today's class is about Immunology Part 5, that is complement system. We'll study about complement system. This class is for BSc MLT third year students. So before we proceed, let me give a short intro about what is complement system. The complement system is a crucial part of the body's innate immune defenses, consisting of about 50 proteins mainly produced by the liver that circulate in blood and tissues in an inactive form. When triggered by infection, tissue injury, or antibodies, these proteins activate sequentially in cascade, amplifying the immune response rapidly. Unlike other immune mechanisms, the complement system can directly kill pathogens by forming membrane attack complexes, mark them for destruction by phagocytosis, that is nothing but opsonization, and recruit inflammatory cells to infection sites. So, studying the complement system is important because it reveals how the body mounts early defense against diverse pathogens even before the adaptive immune response kicks in. It bridges innate and adaptive immunity, helps clear damaged cell, and regulates inflammation. So, understanding complement pathways and regulation also informs research on immune disorders, autoimmune diseases, infections, and novel therapies that manipulate complement for better health outcomes. The complement system exemplifies how enzymatic cascades mediate complex biological functions critical to survival and health. So, let's begin with the introduction. The complement system is one of the most essential part of our immune defense. It forms a bridge between innate system, which provides the body's first line of defense, and the adaptive immune system, which offers more specific protection through antibodies and immune memory. The term complement was originally coined because these proteins complement or enhance the effects of antibodies in destroying pathogens. These complement protein are present in the blood plasma and on the cell membrane, and they work together in a highly coordinated way. When an infection occurs, these proteins interact through a proteolytic cascade, meaning that one protein activates another by cutting it, and the next activates several more, creating a powerful chain reaction. These cascades leads to several immune outcomes like destruction of pathogens, promotion of inflammation, and enhancement of phagocytosis. In simple terms, the complement system acts as a biochemical amplifier of the immune response, ensuring a rapid and effective defense against invading microbes. Next, major functions of the complement system. The complement system serves five major roles in immunity. First and foremost, it can cause lysis of target cells such as bacteria, tumor cells, or enveloped viruses. This happens when the final components of the complement cascade form a membrane attack complex, that is MAC, that creates pores in the cell membrane leading to cell death. Cell death is nothing but lysis of cell. Second, complement generates inflammatory mediators, notably C3A, C5A, which act as chemical attractants called anaphylatoxins that bring immune cells like neutrophils and macrophages to the site of infection. Third complement component like C3B, C4B act as a Opsonins, these molecules coat the surface of pathogens, making them much easier for phagocytic cells to recognize and engulf. Fourth complement helps in clearing immune complexes, preventing their deposition in tissues which could otherwise lead to inflammation or immune damages. Finally, the complement system provides amplification. Once it's activated. Even a small initial immune signal can be multiplied many times over, greatly enhancing the effectiveness of antibodies and other immune components. So, this major function of the complement system is divided into which serves five major roles in immunity. First is lysis of cell, which include bacteria, tumor cells, and enveloped viruses. Second is inflammatory response, generation of peptides C3A and C5A that attract and activate immune cells. Third is opsonization, which is nothing but marking of microbes for phagocytosis, that is C3b and C4b. Fourth is immune complex clearance. It prevents deposition in tissues. And last is amplification. It enhances antibody mediated responses. Next is importance in immunity. The complement system is extremely important because of its speed and efficiency. Unlike antibodies, which take time to be produced after exposure to a new antigen, the complement proteins are preformed in the blood and can act immediately once they detect a pathogen. This gives our body a significant advantage in controlling infections during the early stages before the adaptive immune system is fully activated. Complement also works hand in hand with antibodies. Once they are produced, antibodies can trigger complement activation to destroy pathogens more efficiently. Thus, complement not only provides rapid innate defense but also act as a bridge that connects innate and adaptive immunity, ensuring a seamless immune response that can both recognize and eliminate foreign invaders effectively. So, what is the importance in immunity? It will work immediately upon pathogen detection. It will act before specific immune defenses are activated. It provides rapid and broad spectrum protection, and it bridges innate as well as adaptive immune systems. Next is complement activation. So, overview of complement cascade. Complement activation functions as a biochemical cascade, a series of enzyme driven reactions where the activation of one component triggers the activation of the next, creating a domino effect. Each step amplifies the signal dramatically. For example, when one molecule of C3 convertase is formed, it can cleave and activate hundreds of C3 molecules, resulting in a massive immune response from a small initial trigger. There are about 13 complement proteins directly involved in this cascade. In addition, 7 other proteins regulate or inhibit these reactions, ensuring that complement activation is targeted and does not damage host cells. The concept of cascade is similar to what we see in blood clotting. Once started, it amplifies itself quickly, but it must also be tightly regulated to prevent excessive tissue damage. So complement activation, a cascade is a nothing but a series of enzymatic reactions, which is known as cascade. Each step amplifies the response. Example is C3 to many C3b. It involves 13 main proteins directly in the cascade, as well as 7 other proteins act as a regulators or inhibitors. Next is complement components. The complement system consists of a series of plasma protein named C1 through C9 based on the order of their discovery, not their activation sequence. In the classical pathway, activation follows the sequence C1 to C4 to C2, C3, C5, C6, C7, C7, C8 and C9. When each component is activated, it is split into smaller fragments. The smaller fragment is designated by the lower case letter, that is A, and the larger fragments, which usually remains bound to the complex, is designated B. For example, C3b is cleaved into C3a and C3b. The only exception is C2, where the larger active fragment is known as C2a for a historical reasons. This splitting is key because it generates both enzymatic fragments, which helps activate the next step, and effective fragments which have biological functions such as inflammation, chemotaxis, or opsonization. Next is complement activation pathways. The complement system can be activated through three distinct pathways. First is classical pathway, second is alternative pathway, and third is mannose binding lectin, that is MBL pathway. We'll go one by one. The classical pathway, which is antibody dependent, meaning it requires antigen antibody complexes to trigger activation. Second, alternative pathway, which is antibody independent and can be directly activated by foreign microbial surfaces or endotoxins. Third is mannose binding lectin MBL pathway, which is also antibody independent but is triggered by recognition of specific carbohydrate patterns such as mannose on microbial cell walls. Although these pathways differ in how they start, all three converge at the activation of C3, forming a C3 convertase enzyme. These enzymes plays a pivotal role leading to the formation of C5 convertase and ultimately the membrane attack complex MAC, which causes cell lysis. So in summary, these three activation routes represent different ways the immune system can recognize and destroy pathogens, ensuring flexibility and redundancy in host defense. Next is classical pathway. The classical pathway is the best understood and the first discovered complement activation route. It is initiated by the formation of antigen-antibody complexes, meaning it depends on the adaptive immune system's antibody response. Only specific antibody classes, that is IgM, and certain subclasses of IgG, that is IgG1, IgG2, IgG3, are capable of activating complement via this route. When these antibodies bind to antigens on the pathogen surface, their structure changes, allowing the first complement component, that is C1, to bind to their Fc region, the tail portion of the antibody. Once C1 binds, the cascade proceeds sequentially through C4, C2, C3, and C5, ultimately resulting in the formation of membrane attack complex, that is MAC formation, that punches holes in the pathogen's membrane and causes its death. Diagrammatic representation. In this figure, you will see C1 macromolecular complex consisting of C1Q and two molecules of each C1R and C1S, which is called as 1QR2S2. So what is the C1 complex? This C1 complex is the first protein complex to act in the classical pathway of the complement system, which is a part of our immune defense against infection. It is made up of three main subunits. One, that is C1Q, two C1R, and two C1S. Together, this arrangement is often written as C1QR2S2. So, thus the diagram shows the C1 macromolecular complex, which kickstarts the classical pathway of the complement system. So here that there's a large central C1Q part shown like a stalk or bundle surrounded by enzyme proteins, that is C1R and C1S. C1Q structure and function. C1Q act as a recognition molecule. Structurally, C1Q has 6 arms, collagen like triple helices, forming a bundle of tulip shape with globular heads at the tip. As you can see, heads at the tips. These heads recognize and bind to antibodies like IgG and IgM that have attached themselves to the surface of pathogens. Once C1Q binds to these antibodies, it causes conformational changes or slight shape shifts that lead to activation of C1R. Now, C1R and CS enzyme activation. There are two each of C1R and C1S attached to C1Q. C1R activates itself first, a process called autoactivation, then activates C1S by changing its shape. C1S is a serine protease enzyme, which means it cuts other proteins at a specific sites. C1R and C1S proteins are shaped so that C1R sites at the center with C1S on either end, creating a compact, somewhat eight-shaped assembly. The positioning allows C1R to directly activate C1S when the time comes. So how this activation works in the classical pathway. Once C1S is activated by C1R, C1S starts cutting two other complement proteins, that is C4 and C2. Breaking down these proteins create fragments like C4b and C2b that together form the C3 convertase complex, a key enzyme that amplifies the complement cascade and helps destroy pathogens. Extra functions and flexibility. The C1 complex isn't only for complement activation, it interacts with other molecules like C-reactive protein and pentaxins and can bind directly to the surfaces of bacteria, viruses, or drying cells. Recent research shows that C1Q is very versatile, influencing a variety of cellular processes. So one in whole, C1 is the starter of the classical component pathway composed of C1Q recognition, C1R and C1S enzymes. C1Q binds antibodies, activates C1R, C1R activates C1S, and C1S cuts downstream proteins. These steps amplify our immune response to invading microbes. So, Overview of the complement system, this diagram. The complement system is a group of plasma proteins that helps destroy microbes, enhance phagocytosis, and regulate inflammation as part of the body's innate immune response. Activation of this system occurs through a cascade where each protein is activated in turn, amplifying the immune reaction. Complement activation result in opsonization, recruitment of immune cells to infection sites, and direct lysis of pathogens via the membrane attack complex, that is MAC. So the complement system is activated by three main pathways: classical, that is antibody dependent; alternative, that is microbe driven or no antibodies; and lectin recognizes microbial sugars. So these complement pathway, as I mentioned, the complement pathway is a group of plasma protein that help destroy microbes, enhance phagocytosis, and regulate inflammation as part of the body's immune response. So complement activation result in, as you can see, opsonization and microbial surface, as you can see here, microbial surface and immune complex. So the complement system is activated by these three main pathways: classical, antibody dependent; alternative, that is microbe driven or no antibodies; and lectin recognizes microbial sugars. Each pathway lead to the formation of C3 convertase, which splits C3 protein. The resulting C3b tags pathogens, while C5 convertase C5b leads to cell-killing membrane attack complex formation. Complement fragments also attract immune cells and boost inflammation, helping to clear infection faster. So coming to this slide explanation, so the diagram on this slide represent the complement pathway, showing how the immune system uses a cascade of proteins to target and destroy microbes. This pathway begins when C1 complex composed of C1q, C1r, and C1s binds to antibodies, that is IgG and IgM, attached to pathogens. Activation of C1 leads to the cleavage of proteins, that is C4 and C2, forming what's called the C3 convertase. The C3 convertase splits the complement protein C3 into C3A and C3b, initiating events that mark pathogens for destruction and attract immune cells. Next is alternative pathway. The alternative pathway does not require antibodies. It is triggered when C3b binds directly to microbial surfaces or spontaneously via C3 hydrolysis. Regulatory protein on human cells prevents self-damage, so only microbes are targeted. This pathway also generates its own form of C3 convertase, amplifying the response to infection. Next is lactin pathway. The lactin pathway is activated when mannose binding lactin, that is MBL, binds to sugars, that is mannose residues on pathogen surfaces. MBL then activates MASP enzyme, which cleave C4 and C2, leading to the same C3 convertase found in the classical pathway. So all these three pathways converge at the activation of C3 and the formation of C3b, the opsonin that tags microbes for phagocytosis. Further downstream, complement protein form the membrane attack complex MAC, which creates spores in pathogen membranes, physically destroying them. So in general, the diagram shows multiple ways in which the complement system can be activated through antibodies, direct recognition of microbes, or specialized lectin proteins. regardless of the starting point, all lead to the formation of C3 convertase, which triggers pathogen opsonization, inflammation, and direct microbe cleaning. Understanding these pathways helps explain how the immune system is able to quickly and effectively respond to diverse threats. Next is structure of the C1 complex. So, the C1 complex is a large multi-protein structure composed of C1q, C1r, and C1s. It has the molecular formula C1qr2s2, meaning it contains one Cr. It contains one C1q molecule and two molecules each of Cr1 and C1s. C1q acts as the recognition unit binding directly to the Fc region or portion of the antibody that has attached to the antigen. C1r and C1s serve as enzymatic component responsible for the downstream proteolytic activation of other complement components. The entire C1 complex is stabilized by calcium ions, that is Ca2 plus, which are essential for its structural integrity. You can visualize C1q as a flower-like molecule with 6 arms when it binds to antibodies. It triggers the internal activation of C1r and C1s, setting off the classical pathway cascade. Next is activation steps. Once C1q binds to the Fc region of an antibody that has recognized an antigen, it activates C1r, which in turn activates C1s. The activated C1s then begins a series of proteolytic cleavages. First, C1s cleaves C4 into C4a and C4b. C4b attaches to the microbial surface while C4a diffuses away and acts as an anaphylatoxin, contributing to inflammation. Next is C1s cleaves to C2 into C2a and C2b. The larger C2a fragment binds to C4b to form the enzymes C4b-C2a, which serve as the C3 convertase of the classical pathway. C3 convertase then cleaves the next major component, that is C3 into C3a and C3b. C3a again acts as an anaphylatoxin while C3b binds to the existing complex, that is C4b, C2a, to form C4b, C2a, C3b, which is the C5 convertase. The C5 convertase then cleaves C5 into C5a, a powerful inflammatory mediator, and C5b, which initiates the assembly of the membrane attack complex MAC that will ultimately cause cell lysis. These stepwise sequence illustrate the precision and amplification power of the complement system. Each enzyme activates the next in line, rapidly expanding the immune response. Next is formation of membrane attack complex. After the activation of C5, the complement cascade proceeds to form the membrane attack complex MAC, which is responsible for directly killing the target cell. The process begins when C5b binds to C6, forming the C5b complex. This complex then sequentially recruits C7, C8, and finally multiple C9 molecules. C7 helps anchor the complex to the target membrane, and C8 penetrates the lipid bilayer, creating a site for C9 polymerization. Multiple C9 molecules arrange themselves in circular fashion to form a transmembrane pore. This pore disrupts the integrity of the target cell membrane, leading to the loss of osmotic balance, inflow of water, and ultimately cell lysis. This process is crucial for killing bacteria, especially gram-negative organisms, and infected or abnormal host cells such as tumor or virus-infected cells. Next is non-immunological activators of classical pathway. While the classical pathway is typically antibody dependent, it can also be activated without antibodies. Hence, non-immunological activators exist. Certain microorganisms and viruses can directly activate C1q by interacting with its recognition sites. Examples include some strain of E. coli, that is Escherichia coli, Salmonella, particularly though with low virulence, and viruses such as parainfluenza and HIV. In addition, apoptotic cells undergoing programmed death, expose modified membrane components that can bind C1q, initiating the classical pathway. This mechanism helps the immune system recognize and clear dying or damaged cells, preventing inflammation and autoimmunity. Therefore, these non-antibody triggers broaden the range of complement activation, ensuring defense even before antibody production begins. Next is alternate pathway, alternate, that is pro-Purdin pathway. The alternate pathway functions as an antibody independent defense mechanism, meaning it can activate without any prior sensitization or antibody formation. It is continuously active at a low level in the body, providing a rapid first line defense against pathogen. It gets triggered by pathogen associated molecular pattern, that is PAMPs, such as bacterial cell walls, endotoxins, or foreign particles. This makes it an essential part of innate immunity, allowing the complement system to attack microbes immediately upon entry and before the adaptive immune system is engaged. So this alternative pathway, it is antibody independent activation. It is activated by pathogen surfaces, endotoxin, or any foreign particles, and it is important first line defense of mechanism. Membrane attack complex, MAC. This is a schematic representation of terminal or membrane attack complex of complement. The MAC forms pore on the surface of microbes causing lysis. Now, what is MAC? The membrane attack complex is a structure formed by the terminal complement pathway, a part of our body innate immune defense. It consists of proteins, that is C5b, C6, C7, C8, and many molecules of C9. MAC's function is to punch holes in pathogen cell membrane, leading to their cell lysis and death. So step-by-step assembly. First, activation begins. All three components pathway, that is classical, alternative, and lactin, converge to form a C5 convertase. This enzyme cleaves C5 into C5a, an inflammatory signaling molecule, and C5b, the scaffold for MAC assembly. Second is protein binding sequence. C5b binds C6 and then C7, making C5b 7 capable of associating with the cell membrane. Membrane binding C8 attaches, causing C5b to C8 to begin penetrating the lipid bilayer. And last is pore formation. Multiple copies of C9 up to 18 are recruited and form a ring-shaped pore in the membrane. This structure allows water, ions, and small molecules to flow freely, leading to osmotic imbalance and cell death. So in this diagram, you'll see a layered assembly C5b at the center, sequentially surrounded by C6, C7, C8, and a large ring of C9 units. So, arrows or steps may depict how each protein joins the complex and how the final structure embeds in the pathogen's membrane. Pores, that is nothing but holes, are visible in the membrane, indicating loss of integrity and cell lysis. So MAC is specially potent against gram-negative bacteria but can lyse any cell-lacking protein inhibitors. Human cell-expressed regulatory proteins like clusterin or others to prevent MSC from forming on themselves. Sometimes incomplete MSCs don't insert into membranes, remaining inactive in fluid and scavenged by stabilizing proteins. So before cell lysis, MSC can trigger signaling inside host cells such as inflammation if the pore is not lethal. Next is activators of alternative pathway. The alternative pathway can be activated by various microbial and non-microbial substances. Among the most common activators are bacterial endotoxin (LPS) from gram-negative bacteria, which directly trigger complement activation. Some antibodies such as IgA and IgD can also activate this pathway in specific conditions. Additionally, cobra venom factor, a component from cobra venom, mimics part of the complement system and continuously activates the alternative pathway. Another notable activator is the nephritic factor, an autoantibody found in certain kidney diseases like membranoproliferative glomerulonephritis, which stabilizes complement convertases and leads to excessive activation and tissue damage. Key components. The alternative pathway does not use the early components, that is C1, C2 or C4 seen in the classical pathway. Instead, it relies on a unique set of plasma proteins, that is C3, factor B, factor D, and properdin, that is factor P. C3A is the central molecule of the complement system and can undergo spontaneous hydrolysis to initiate the cascade. Factor B binds to C3b, forming a complex that is cleaved by factor D into BB, which remains attached to C3b to form C3b, B and B, the C3 convertase of the alternative pathway. Properdin, that is factor P, stabilizes this enzyme complex, increasing its half-life and activity. Together, these factors enable efficient amplification of complement activation on pathogen surfaces. Next is mechanism. So the alternative pathway is initiated by spontaneous hydrolysis of C3 in plasma, forming C3b. This C3b molecule attaches to microbial surfaces where it binds factor B. Factor D, a serine protease, then cleaves factor B into B A and BB, generating the C3b-BB complex, which act as the C3 convertase. This enzyme cleaves more C3 into C3A and C3b, amplifying the process. When another C3b binds to the C3 BBB complex, it forms C3B, BBC3B, the C5 convertase, which cleaves C5 into C5A and C5B. C5B initiates the formation of the MAC, that is C5B and C9, leading to cell lysis. Properdin plays a vital role by stabilizing these complexes and ensuring strong complement activation on pathogen surfaces while preventing activation on host cell. Next is mannose binding lactin, that is MBL pathway. MBL pathway is another antibody independent complement activation route. It is targeted or it is triggered by carbohydrate recognition, specifically when MBL, a pattern recognition molecule, binds to mannose residues on the surface of pathogens. This pathway is particularly important in recognizing and defending against organisms such as Listeria, Salmonella, and Candida albicans. Since mannose is rarely found on mammalian cells but is abundant on microbial surfaces, this pathway allows the immune system to distinguish self from non-self. Mechanism of MBL. When MBL binds to mannose residue on microbial surfaces, it forms a complex with MBL-associated serine proteases. That is MASPs, mainly MASP1 and MASP2. These proteases mimic the actions of C1R and C1S from the classical pathway. MASPs cleave C4, C2 to form C4B, C2A, which acts as the C3 convertase, leading to C3 cleavage. This produces C3B, which then participates in C5 convertase formation and ultimately in MAC assembly, that is C5B and C9. Hence, the MBL pathway follows a mechanism similar to the classical pathway but differs in its activation trigger, carbohydrate recognition rather than antigen-antibody interaction. This schematic representation of figure mannose binding lectin, that is MBL pathway of complement activation. MBL bound to mannose containing residues CHO interact with MBL activated serine proteases, that is MASP. Activation of MASP leads to activation of components C2, C4 and C3. So what is the lactin pathway? The lactin pathway activates the complement system in response to certain carbohydrates present on the surface of microbes. It does not require antibodies and is a key part of innate immunity, offering early and rapid response against infection. Mechanism and components. Pattern recognition, MASP, activation, C3 convertase formation, and downstream event. So these four things is important in mechanism and components. So you will see MBL and MASP's binding to microbial surface sugars followed by a cascade reaction where C2 and C4 are cleaved and C3 convertase is formed. Interaction with microbial carbohydrates drive recognition and enzymatic activity. The cascade parallels the classical pathway but starts without antibodies, demonstrating the power of innate immunity. So, this diagram highlights the lactin pathway, an antibody-independent route of complement activation, mannose binding lactin MBL and Ficolin sends sugars on microbes and recruit MASP enzymes. MBL attaches to a pathogen, that is MASP1 is activated and in turn activates MASP2. MASP2 breaks apart C4 and C2, allowing C3 convertase to form and trigger the complement cascade, a rapid response to infection. Formation of C3b tags microbes for phagocytosis, while downstream assembly of the MAC enables direct microbial destruction. The lactin pathway shows how our immune system can sense and destroy pathogens even before antibody production begins. Regulation of complement system. Now, need for regulation. The complement system, while protective, can be dangerous if it left uncontrolled, as it has the potential to damage the body's own cells. Therefore, precise regulatory mechanisms are essential to ensure complement activation occurs only on foreign surfaces. The body employs several methods for control. The short half-life of active components, that is complement fragments, ensures they do not persist long enough to harm host tissues. Second, multiple regulatory proteins in a plasma and on cell membranes inhibit inappropriate activation, maintaining immune balance and preventing disorders like autoimmune hemolysis or tissue inflammation. Next is stages of regulation. Complement regulation occurs at three main stages during the cascade. One, before assembly of convertases, two, after assembly of convertases, and three, during MAC formation. So first, before convertase formation, that is preventing early activation of the pathway. Second, after convertase assembly, controlling amplification and progression. Third, during MSC formation, preventing attack on self-cell membranes. This multi-level regulation ensures the complement system remains efficient in killing pathogens while protecting host cells from accidental injury. Next is regulation before convertase formation. Several key regulatory proteins control complement activation at the pre-convertase stage. First, C1 inhibitor C1INH prevents the activation of C1R and C1S in the classical pathway, stopping the cascade at the earliest point. Second, C4b binding protein. C4b BP binds C4b and prevents its association with C2A, thereby blocking C3 convertase formation. CR1 complement receptor 1 and MCP membrane cofactor protein act on cell surfaces to prevent formation of the C4b C2A complex. Factor 1, a serine protease that inactivates C3b and C4b by cleaving them into IC3b and C4d, ensuring they cannot form convertases. Factor H competes with factor B for binding to C3b, preventing C3 convertase formation in the alternative pathway. Together, these mechanisms protect host cells from complement-mediated injury while maintaining effective immunity against pathogens. Next is regulation after convertase assembly. Once the complement convertases C3 and C5 convertases are formed, the system must still be tightly regulated to prevent excessive or prolong activation. Several regulatory protein functions at this stage to dissociate or destabilize convertase complexes. So C4b binding protein, that is C4b BP, CR1, complement receptor 1, factor H, and DAF, that is DK accelerating factor, act by accelerating the dissociation of C2A or BB from the C3 convertase, effectively halting further complement activation. In contrast, properdin factor P serves the opposite role. It stabilizes the C3 convertase of the alternative pathway, that is C3BBB, prolonging its activity on microbial surface while ensuring it does not persist on host cell. Additionally, anaphylatoxin inactivator, also known as carboxypeptidase B, regulates inflammation by degrading potent inflammatory peptides C3A, C4A, and C5A, preventing tissue damage due to overactive inflammation. These mechanisms helps keeps the system balanced between effective defense and protection of self tissues. Next is regulation at MAC formation. The membrane attack complex MAC, if uncontrolled, could damage host membranes. Hence, its formation is also regulated. The S protein, this S protein prevents the C5b67 complex from inserting into host cell membrane, thereby halting MAC assembly before it begins. Two other key regulators, HRF, homologous restriction factor, and CD59, also known as membrane inhibitor of reactive lysis, MIRL, act at the final step of the cascade. They inhibit the addition of C9 to the forming MAC complex, preventing pore formation and cell lysis. This ensures that complement-mediated killing occurs only on target cells and not on normal host cell, preventing cellular integrity while maintaining immune efficiency. Next is biological consequences of complement activation. Activation of the complement system leads to a wide range of biological outcomes that are crucial to both innate and adaptive immunity. These include cell lysis, inflammation, enhanced phagocytosis, immune complex clearance, and virus neutralization. These outcomes work together to eliminate pathogens quickly and efficiently while also promoting tissue healing and immune regulation. Thus, complement activation is not only a killing mechanism but also a bridge that coordinates multiple immune processes. So in this complement binding receptor table, so we'll explain everything one by one. What are complement receptors? Complement receptors, CRs, are protein expressed on the surface of various immune cells that recognize and bind complement fragments deposited on pathogens or immune complexes. They facilitate pathogen clearance, immune cells activation, and link innate and adaptive immunity. So how complement receptor work? These receptors bind complement fragments such as C3b or its degradation products, IC3b, C3d, on pathogens or immune complexes. Binding promotes opsonization, enhancing phagocytosis by macrophages and neutrophils. Some receptors, like CR2, enhance B cell responses by providing signals that augment antibody production. CR1 also helps regulate complement activation by aiding in degradation of complement components to prevent excessive inflammation. So what it looks like, so various immune cells expressing CR1, CR2, CR3, and CR4. Complement fragments, example C3b, IC3b, opsonizing a microbe, and interaction of complement receptors with these fragments leading to phagocytosis or immune activation. So, complement receptors are key proteins on immune cell that recognize fragments of the complement proteins, most commonly C3b and its breakdown products. CR1 is widely found on immune and other cells and helps clear immune complexes, regulate complement activity, and promote phagocytosis. CR2 is mainly on B cell and follicular dendritic cells, where it aids in enhancing antibody responses by acting as a co-receptor. CR3 and CR4 are integrin type receptors that bind IC3b. They mediate adherence to and engulfment of complement-coated pathogens, critical for innate immunity. These receptors bridge complement activation with immune cell recruitment and pathogen elimination, highlighting complement's role beyond protein cleavage to immune modulation. Next is major biological effects. Complement fragments perform specific functions that lead to several major immune effects. Opsonization. Fragments such as C3b and C4b bind to pathogen surfaces, marking them for phagocytosis by macrophages and neutrophils. Next is chemotaxis. C3a and C5a act as the chemical attractants, drawing immune cells like neutrophils and macrophages to the site of infection. Third, inflammation. Fragments C3a, C4a, and C5a trigger mast cells and basophils to release inflammatory mediators. Anaphylotoxin activity. These same peptide act as anaphylotoxins, stimulating mast cell degranulation and histamine release, leading to vasodilation and smooth muscle contraction. Increased vascular permeability. C2 kinine, another complement product, contributes to increased permeability, allowing immune cells and plasma proteins to access infection sites. Together, these effects promote a strong and localized inflammatory response to help contain and destroy pathogens. Next is major biological effects of the complement system. The complement system is a crucial part of the innate immune system that rapidly responds to infection. Its activation results in several biological effects that work together to clear pathogens and modulate immune responses. These effects include pathogen lysis, opsonization, inflammation, and immune cell recruitment. So, a biological effect illustrated in this slide. First, direct cell lysis via membrane attack complex MAC. Second, opsonization and enhanced phagocytosis. Third, generation of inflammatory mediators. Fourth, recruitment and activation of immune cells. Fifth, role in adaptive immunity. And sixth, clearance of immune complexes and apoptic cells. So what the diagram shows, multiple cascade events from classical and alternative pathways leading to complement activation. Opsonized microbes being engulfed by phagocytes via complement receptors. Formation of the MAC causing pathogen lysis. Release of small fragments inducing inflammation and interaction with B cell enhancing humoral immunity. So, this slide summarizes the broad biological effects of the complement system, illustrating its crucial role in defending the host from infections. First, the formation of the membrane attack complex physically disrupt the membrane of microbes, causing their death Second, opsonization. Opsonization tagged these pathogens, making them more attractive target for immune cells like macrophages and neutrophils. Third, small complement fragment act as inflammatory mediators to recruit and activate immune cells, amplifying the immune response. Fourth, complement bridge innate and adaptive immunity by helping in antigen presentation and enhancing B cell antibody response. And fifth, lastly, complement aid in clearing immune complexes and apoptic cells, which is vital for preventing autoimmune reactions and maintaining health. Other important effects: in addition to the primary functions, complement activation produces several specialized immune effects. Phagocytic enhancement occurs when complement fragments like C3b, C5b coat microbes, making them easier for phagocytes to recognize and ingest. Phagocytic enhancement occurs. So next is virus neutralization is another critical function. For example, complement working together with IgM antibodies can neutralize viruses such as herpes virus, preventing them from entering host cell. Cytolysis mediated by the MSC C5b to C9 leads to the direct destruction of target cells, including erythrocytes, bacteria, and tumor cells. Lastly, immune adherence refers to complement-coated immune complexes binding to red blood cells via complement receptors. These mechanisms transport complexes to the liver or spleen for safe removal, preventing their deposition in tissues and potential inflammation. Next is complement and disease. While complement is protective, dysregulation or inappropriate activation can cause disease. In type 2 hypersensitivity reactions, complement participate in cytotoxic responses, leading to cell lysis in conditions like hemolytic anemia. In type 3 hypersensitivity, complement contributes to immune complex diseases such as serum sickness and systemic lupus erythematosus SLE by amplifying inflammation around immune complex deposits. Autoimmune diseases like SLE and autoimmune hemolytic anemia often involve excessive complement activation and tissue injury. Overactivation of the alternative pathway in endotoxic shock caused by bacterial endotoxin can trigger disseminated intravascular coagulation, that is DIC, thrombocytopenia, and severe inflammation. Hence, controlled complement activity is essential to avoid self-damage. Next is defects in the complement system. Complement deficiency can affect both innate and adaptive immunity. Since complement bridges these two systems, such deficiency typically result in recurrent bacterial infection, poor immune complex clearance, and autoimmune manifestation. Depending on which component is lacking, the outcome may involve increased susceptibility to certain bacteria, impaired inflammation, or autoimmune tissue injury. Thus, complement deficiencies are important clinical conditions that can reveal much about immune functions. Early component deficiencies. Deficiency in early classical pathway components C1q, C1R, C1S, C4, or C2 often lead to defective immune complex clearance. This causes immune complex disease such as SLE, vasculitis, as the body fails to remove antigen antibody complexes efficiently. Additionally, patients with these deficiencies are more prone to recurrent pyogenic infections, particularly from Staphylococcus and Streptococcus species due to poor opsonization and reduced complement-mediated killing. C3 deficiency. C3 is the central and most critical complement component shared by all three activation pathways. A deficiency in C3 result in severe impairment of the entire system. Without C3, opsonization, chemotaxis, and MSC formation are all affected, leading to recurrent bacterial infection, especially with encapsulated organisms such as Neisseria, Streptococcus pneumonia, and Haemophilus influenza. C3 deficiency also causes immune complex diseases as clearance of immune complexes is dependent on C3b mediated binding to red blood cells for removal. Thus patients show both infectious and autoimmune complications. Next is alternative pathway deficiency. Deficiency in factor D or properdin factor P impair the alternative pathway's ability to form stable C3 convertases. This leads to an increased susceptibility to Neisseria infections, including meningococcal and gonococcal diseases, because the alternative pathway provides rapid antibody independent responses. Its deficiencies leaves individuals with delayed or inadequate bacterial killing, leading to recurrent infections, particularly of the respiratory tract. Next is terminal component deficiency. The terminal components C5 to C9 are responsible for forming the MAC formation. Deficiency in any of this component result in defective cell lysis. Patients with this deficiency are particularly prone to recurrent infections by Neisseria meningitis and Neisseria gonorrhea, that is meningococcal or gonococcal infections, because these bacteria are normally killed through MSC mediated lysis. Interestingly, immune complex clearance remains intact since earlier complement functions like C3 activation are unaffected, meaning these patients typically don't develop autoimmune systems. Next is regulatory protein deficiency. Defects in complement regulatory protein cause uncontrolled activation leading to tissue injury. A deficiency of C1 inhibitor C1INH result in hereditary angioneurotic edema, that is HAE, a condition marked by excessive inflammation, tissue swelling, and airway obstruction due to uncontrolled activation of C1 and bradykinin production. Deficiency of DAF, that is decay accelerating factor, and CD59 causes paroxysmal nocturnal hemoglobinuria, that is PNH. This disease in PNH red blood cells lack this protective surface protein and are destroyed by complement mediated lysis, leading to hemolytic anemia, hemoglobinuria, and thrombosis. Next is synthesis of complement. Site of complement synthesis. In complement proteins are mainly synthesized by several organs and cell types. The liver is the primary site for production of most complement components found in plasma. The spleen contributes to complement synthesis during immune responses, producing certain regulatory and activation proteins. In addition, phagocytic cells such as macrophages and monocytes can produce complement proteins locally at infection sites, providing rapid reinforcement of immune defenses directly at the site of pathogen invasion. So this side of complement synthesis, three types: liver, spleen, and phagocytic cells. Liver, it is a major site for plasma complement proteins. Spleen, it contributes to immune complement synthesis. Phagocytic cells, it synthesizes complement locally at infection site. Next is Summary of this class. So, in summary, the complement system serves as a vital link between innate and adaptive immunity, coordinating defense mechanisms that act rapidly and specifically. Activation occurs through three main pathways: classical, alternative, and MBL, all of which converge on a common terminal sequence that leads to MSC formation and pathogen destruction. Its activity is tightly regulated to prevent host cell damage, ensuring a balanced and controlled immune response. Complement plays a major role in host defense, inflammation, phagocytosis, and immune regulation, while its deficiency or dysregulation can lead to infections, autoimmune diseases, or inflammatory disorders. So, understanding this system provides a foundation for appreciating many immunological diseases and the therapeutic use of complement inhibitors. So overview of and introduction of summary. The complement system is a key component of both innate as well as adaptive immunity. The term complement refers to its role in enhancing antibody activity. It is composed of plasma and membrane-bound protein that circulate in an inactive form. Activation will occur through a proteolytic enzyme cascade where one protein activates the next. Major roles in pathogen lysis, inflammation, opsonization, immune complex clearance, and immune amplification. It acts as a biochemical amplifier of immune defense and provides immediate protection. Functions and importance: cell lysis via formation of the MSC, inflammation through release of anaphylatoxins through C3A and C5A, opsonization C3 and C4B coat microbes to enhance phagocytosis, immune complex clearance, it prevents immune complex diseases, bridges innate and adaptive immunity, acting synergetically with antibodies, and rapidly mobilized since most components are already present in plasma. Complement cascade, it operates as a sequential enzymatic chain reaction similar to blood clotting. One activated component can activate hundreds of downstream molecules, that is amplification effect. It contains 13 core, around 13 core complement proteins and several regulatory proteins. Its main component named C1 to C9, not in order of activation. Classical activation order is C1 to C4, C2, C3, C5 to C9. Cleavage produces fragments, smaller is A and larger is B subunits, except C2 where C2A is larger. Pathway of activation, there are three distinct activation pathways, all converging at C3 activation. Classical pathway, it is antibody dependent. Alternative pathway, it is antibody independent. Microbial surfaces triggered. MBL, that is mannose binding lactate pathway, it activated by mannose residues on pathogens, all culminate in C5 convertase formation to C5 cleavage to MSC, that is C5B to C9, and then cell lysis. Classical pathway trigger is antigen antibody complex, mainly IgM, IgG1, IgG2, and IgG3. C1 complex is equals to C1q recognition as well as C1R and C1S, that is together is enzyme. C1 activation requires Ca2 plus ions for stability. Sequence of activations: MSC assembly, effective mainly against gram-negative bacteria and virus-infected cells. Some bacteria, viruses, and apoptic cells can directly activate C1q as non-immunological activators. Second is alternative pathway. It is a part of innate immunity and does not require antibodies. Continuously active at a low tick over level via spontaneous C3 hydrolysis. It is activated by bacterial surfaces, endotoxins, LPS, zymosin, IgA, or cobra venom factor. Components include C3, factor B, factor D, and properdin, that is factor P. Mechanism is this: it provides immediate defense before antibodies appear. Nephritic factor can stabilize convertase abnormally to chronic activation and tissue damage. Next is MBL, mannose binding lactin pathway. It is triggered by MBL binding to mannose residue on microbial surfaces. It detects mannose-rich microbes, that is bacteria, fungi, yeast-like candida, all these microbes, distinguishing them from host cell, that is mannose-poor. MASP1 and 2 cleave C4 and C2 and form this. The cascade then parallels the classical pathway and which will lead to MSC formation. Entirely antibody independent, important in early innate response. Regulation of complement activity. Complement activation must be tightly controlled to avoid self-cell damage. It is controlled through short half-life of activated fragments or regulatory proteins in plasma or on host cell membranes. Regulation occurs at three stages: before convertase formation, that is prevention of activation; after convertase assembly, that is control of amplification; and during MSC formation, that is terminal stage of control. So before convertase formation, as you can see, all this, and after convertase assembly, this. Biological consequences. Complement activation produces several major outcomes, that is cell lysis, opsonization, inflammation, chemotaxis, increased vascular permeability, immune adherence, and virus neutralization. Next is clinical correlation and disease roles. Overactivation or deficiency can lead to disease such as type 2 hypersensitivity and type 3 hypersensitivity, autoimmune disease, and endotoxic shock. Complement deficiency, it detects it defects can affect innate as well as adaptive immune defenses, causing recurrent bacterial infection or autoimmunity. Early component deficiency, that is C1, C4, and C2, it is impaired immune complex clearance through this. C3 deficiency, that is central molecule missing, all pathways are impaired. It will lead to severe bacterial infection including Neisseria, Streptococcus, and Haemophilus. It causes defective opsonization, chemotaxis, immune complex clearance. Next is alternate pathway deficiency, factor D or properdin deficiency. It is weakened alternative activation. It is prone to Neisseria, meningitis, and gonorrhea. Its terminal component deficiency include such as C5 to C9. It is showing impaired MSC formation, which will lead to defective lysis and recurrent Neisseria infection, immune complex clearance unaffected, and regulatory protein deficiency gives C1 IHN deficiency, hereditary HAE, that is swelling of skin, gut, and airway, DAF or CD59 deficiency, that is PNH, and complement mediated RBC lysis, anemia, and thrombosis. So complement synthesis, main site is liver, others are spleen, macrophages, and monocyte. Local synthesis at infection site enhance targeted immune response. So overall summary, the complement system forms a vital link between innate and adaptive immunity. There is three activation pathways: classical, active, and MBL converge to produce the MSC, leading to pathogen lysis. Strict regulatory control ensures self-protection. Functions include defense, inflammation, opsonization, immune clearance, and regulation. Deficiency or dysregulation results in infections, autoimmune infections, or inflammatory diseases. Understanding complement mechanism is crucial for immunology, diagnostics, and therapeutic applications. So, question parts for 3 marks. First, name the main regulatory protein that dissociate complement convertases. So here you can name C4BBB, C4BBP, CR1, factor H, and DAF. Second, what is the role of properdin, factor P? It stabilizes the alternative pathway C3 convertase, that is C3BBB, prolonging its activity. Third, mention two regulators that prevents MSC formation. So S protein, that is vitronectin, and CD59, that is MIRL. These are the two regulators that prevent MSC formation. Fourth, list three main biological outcomes of complement activation. That is cell lysis, inflammation, and opsonization are the three main biological outcomes. Fifth, which complement fragment act as anaphylatoxins? So that is C3A, C4A, and C5A. These are the complement fragment which act as this. Sixth, what causes hereditary angioneurotic edema? That is HAE. Deficiency of C1 inhibitor, that is C1-INH, will causes hereditary angioneurotic edema. 7. What infections are common in terminal component, that is C5 to C9 deficiency? Recurrent Neisseria meningitis and Neisseria gonorrhea infection are common in terminal component of C5 to C9 deficiency. Eighth, which component deficiency affects all pathway? C3 deficiency will affect all pathway deficiency. Ninth, what is the function of the S protein in complement regulation? Prevent insertion of the C5b67 complex into host membranes. This is the function of the S protein in complement regulation. 10th, name three main sites of complement synthesis. Three main site of complement synthesis is liver, spleen, and phagocytosis cell. Questions for 5 mark. First, explain how complement activation is regulated after convertase assembly. Second, describe the regulation of MAC formation. Third, list and explain the major biological effects of complement activation. Fourth, write short notes on other important effects of complement. Fifth, explain the role of complement in disease development. Sixth, discuss the effects of early component deficiencies of the complement system. 7. Describe C3 deficiency and its consequences. 8. Write a note on alternative and terminal component deficiencies. 9. Explain regulatory protein deficiency and their clinical importance. 10. Give an account of complement synthesis and its immunological significance. So with this, we completed this complement system topic. So that's all for today's class. I hope all will understand the concept of complement system as I explained everything in detail from introduction till end. So that's all for today's class. Thank you.